Hello, everyone, and welcome to the next chapter, which is called Host versus Virus. So, here we're going to be talking a lot about how our host is able to fight viral infections. So, very specifically going into viruses, and we'll definitely be talking a lot about this, specifically more the innate immune system, because the adaptive immune system is quite, let's say, specialized towards not specialized, it's more like you see it very commonly between bacterial infections and viruses. What really differs a lot is the innate immune response. So we'll definitely be diving a lot today on the innate immune response and how it's able to control viruses. So as I said, we all of us know here that well, I kind of hope that our immune system is divided into two parts: the innate immunity and the adaptive immunity. Other than of course, they differ in the time. They differ a lot on and in a very important evolutionary aspect in how they recognize certain pathogens. So we know that the innate immune system is not so specific. It actually is not specific towards certain microorganisms, but rather they look at more, let's say, patterns. They look at PAMPs. These are pattern associated molecular patterns, basically, or like pathogen associated molecular patterns. And they do this with their PRRs. These are pathogen recognition receptors. So they're able to recognize, let's say, certain patterns that we see a lot within pathogens. This could be sugars, this could be certain ways DNA or RNA are handled, basically, if they're double stranded, if they're single stranded, antisense, many different things. This is how the innate system is able to detect. While our adaptive is more specialized. What happens here is you're trying to educate certain cells like your B or T cells to recognize certain epitopes that you are exposed to during an infection. That is more your adaptive immune system. So, despite them being very, very different, they're very interconnected, in my opinion. That the innate immune system really teaches, it's the one that teaches your adaptive immune system. So, it's not that one of the others is important or not. Your innate immune system is us. If we have no B or T cells, we die. This is called severe combined immunodeficiencies that we spoke about. So it shows to see how important our adaptive immune system is. But if you also have no innate immune system, well, your adaptive really gets doesn't get taught. It's the one that connects. So both are, let's say, indispensable and interconnected that are super important to actually focus. And one thing that I realized a lot during my research or throughout my years in research, a lot of people were really focusing on just the innate or just the adaptive immune system. No one really, well, there of course there were people, but very rarely did you see researchers trying to interconnect them together. And this is what I started to realize, especially specifically in the lab that I work in. In my lab, a lot of people focus on adaptive immune system. Very little. I think I'm the only one in my lab. Now there's another person, it's a PhD under with me, that we both start to more look at the innate immune system. But we try to look on how the innate immune system can be used to make your adaptive immune system much, much better in cancer because they're so interlocked. So think about the innate immune system, not just purely from a sake of okay, it acts super quickly because it doesn't need to be taught. It looks at pattern recognitions, these PAMPs, and they do this using basically their PRRs, and then they basically are literally trying to combat your viral infections. But think about them also as that buying time. They like to buy time because, as we know, the adaptive immune system uh, takes some time to actually be formed. So what happens is that your innate immune system is the first, let's say, mechanism that is really trying to first prevent viral infections to occur. So it's basically trying to help everything for at the beginning, and then at the same time, it's also teaching your adaptive immune system to come in later. So think about that there. But just to remember that the innate control is extremely important because without it, specifically in a viral infection, your viral replication goes overwhelmingly. It increases to the point where your adaptive immune system has no chance in stopping it. So it's both of them are interlocked. So having an innate immune system, it really kind of damps it down, and then it teaches your adaptive immune system to come in and do the rest of the work. So if you don't have your innate immune system, your viral replication is just ramped up. So, which to the point that despite your adaptive immune system being able to kill it, it's just too much. It's too overwhelming for it. And again, if you don't have adaptive immune system, your innate is not able to by itself remove it. So again, this is a whole between your innate and your adaptive immune system. There's a temporal dynamics. So where we see at the beginning our innate immune system kicking in. So we see your innate immune response is the first one. So these are days after infection. So between days 0 and let's say 5, you see basically a lot of innate immune responses going literally up. So what happens in that kind of sense from the innate immune response is, in general, you have a lot of them coming up trying to reduce your viral titers. But then, of course, comes later on your adaptive immune response. These are your cytotoxic T cells, your antibodies. These are where your B and T cells start to kick in. So it's usually actually your innate immune response, specifically certain cytokines, certain chemokines that are basically shaping up our adaptive immune responses. And of course, this is why we have the first coming up of your innate. And we really have to, what we really see, if we look at the more temporal dynamics, you have these interferons. These are different types that we'll be talking about that come in, and what they really do are trying to stop your viral titer. They also kickstart your NK cells, which then come in to start literally getting activated, and you start bringing other immune cells like your monocytes or macrophages. They start to come in, and what they start doing is producing a lot of different cytokines and chemokines that are shaping up your cytotoxic T cells and your antibody titers, so your B cells. So one thing that you start to see really, specifically here, your cytotoxic T cells are extremely important because they're the ones that as soon as they start coming up, you really start to see your antibody titer going down. So think about your innate immune system. It's just stopping your, let's say, this from going way too high. So it's really dampening down, because without these, this viral titer would be extremely high. But it's not able to really fully control it. It's your cytotoxic T cells that come in to really fully control this. Your antibody titer comes a bit later, and to the point that where it's at its highest, your viral titer is almost gone. Because think about viral titers or antibody titers in a specific sense that antibodies are there to protect you. They're really there to stop viruses from literally going from cell to cell. Antibodies, as we know, they do not go inside a cell. So viruses are inside the cell, so antibodies are useless when it comes to killing virally infected cells. That is the job of the cytotoxic T cells. So we see a lot, in generally, we kind of use antibody titers a lot as a protective, let's say, correlation. So if someone has high amounts of antibodies, we know that this person is protected, because the viruses now, when they're trying to go from cell to cell, they're not going to be able to, because the antibodies are going to be neutralized. So we use antibody titers a lot in some sort of way as a protective. So as we said, it's really a lot the cytokines and the chemokines coming from your innate responses that are really, let's say, helping us in determining the magnitude and phenotype of this adaptive immunity. So it really shapes up our adaptive immune responses. But sometimes what happens is that too much innate responses, like when you have, let's say, chronic viral infections, so these are the ones that are really going for long times, or hyper acute, meaning extremely high activation of your innate immune system. This can be very toxic. You get something called cytokine storm syndromes. These are literally a storm of cytokines that are, other than, of course, that shaping up your adaptive immune response, they're causing a lot of damage towards your surrounding cells, and this can be extremely dangerous. A pure example of this is Ebola. You get infected with Ebola, this could really be detrimental to your body. So now I want to talk about certain cytokines and how they're really important within your adaptive immune responses, and generally how they fight viral infections. And the most important ones that come with viral infections are interferons. There's three main types. There is the type 1, which is alpha and beta, type 2, which is gamma, and type 3, which is lambda. Type 1 are mostly produced by almost every nucleated cell, so they're produced by almost every cell that can be infected by a virus. So that's why they need to be produced. And what they do is they really are responsible in kind of sounding the alarms in your system, and it kind of puts cells in this antiviral state. What that means, we're really going to go into, let's say, more definition about that. Type 2 is more different. It actually is produced mostly by your activated innate cells, or basically adaptive cells, specifically activated T cells and natural killer cells. And what they do is once your activated natural killer cells or T cells are activated, they start to release interferon 2, type 2s. So these are interferon gamma. And interferon gamma, it's a huge or very important cytokine that really kind of makes macrophages much more activated, but also makes other cells start to do antigen presentation. So it really pushes MHC class 1 and MHC class 2 presentation, extremely, extremely important. So it's trying to adapt your innate and adaptive immune system. Think about type 2 interferons like interferon gamma as just literally trying to connect your innate and adaptive by making active macrophages activated, because macrophages also do antigen presentation, but also they do a lot of, they promote antigen presentation as well. Type 3 is the lambda interferon. They're very similar to type 1, but type 1 works more systemically. Type 2 only mostly works in the gastrointestinal and respiratory tracts. These are your epithelial kind of linings, where basically what they do is they really limit viral replication, and they reduce basically, they try to reduce basically any viruses from leaving your mucosal sites in your gastrointestinal and respiratory tracts. So think about that in a sense. So type 1 and type 3, the difference is that type 1 is more systemic, type 3 is more protection at the mucosal basically surfaces. So type 1 and interferons, which are extremely important when it comes to viral infections, these are alpha and beta. And as I said, they can be produced by all nucleated cells, because all nucleated cells can be infected by viral infections. So the most important thing is that these, when basically they are, when we have a viral, or when we have basically a normal cell will, once it's, let's say, infected by a virus, it's going to release these type 1 interferons. And type 1 interferons, they actually work in a, let's say, autocrine and paracrine signaling, meaning that once produced, they can be basically produced up, and they can literally bind to their own receptor produced from their own cell, hence autocrine, to basically induce a signaling. But they can also be paracrine, meaning that they can also bind to other receptors found in other cells. So this is very important. Once bound to their very important receptors, this induces in some sort of way a signaling cascade, which is specifically what it does, it really pushes quite a lot these interferon stimulated genes, these ISGs. So ISGs, what they do is they technically, these are proteins, these are interferon stimulated genes, these are proteins that are produced that literally disrupt multiple stages of the viral cycle, life cycle. So one very important thing that you need to remember with type 1 interferons is that they do not kill directly viruses. They do not do that. What they do is they stop their replication. They stimulate these ISGs, these interferon stimulated genes, to produce proteins that are messing up with viral life cycles. And we're going to be talking specifically what that means. But this could be, for example, going down to stopping replication, basically, of the viruses. This could be stopping the genome replication. This could be stopping basically the RNA production, so protein synthesis, and all in that. But this is not specific. So when they do stop DNA replication, or when they do stop protein synthesis, they stop it for both the host cell, but also the viruses. So it's kind of a trade-off, meaning that when these, let's say, ISGs are turned on, they will stop the host from replicating its genome or its proteins, and it will stop also the viral genome and viral proteins to replicate. So it's a trade-off. You're sure stopping your viruses from, let's say, pushing up, but they also stop cells from replicating, your basically host cells. So what happens technically, as we said, your type 1 interferons, they usually are triggered by certain viral components, particularly, specifically, I would say your double-stranded RNA, because no matter what, let's say the majority of cells, even they come of DNA origin or of single cell origin or double-stranded origin, they will always, not always, but majority of the cases, will be giving you an intermediate that is double-stranded RNA. And double-stranded RNA is a very heavy PAMP, because we do not have double-stranded RNA. So these are usually recognized by PRRs. They really are recognized by them, and what these PRRs will do is they literally will start a signaling cascade. So of course, double-stranded RNA is not the only PAMP, but this could be, of course, in single-stranded RNA or DNA that is heavily CPG methylated. So these CPG are basically are very CPG islands are extremely big islands that have a lot of C and Gs in the DNA. These are coming from certain DNA viruses that can also cause PRRs to be activated. So the activation of PRRs really pushes these ISGs, really basically all this kind of stuff. Oh, so sorry, my bad. The activation of these PRRs leads to the activation of type 1 interferons. And type 1 interferons, what they do is they really will go out and basically, these type 1 interferons will go and activate the ISG productions or stimulation of ISGs to produce the proteins that will stop in some sort of way, let's say, your viruses from replicating. So, as we said, the host cells that detect through pattern recognition receptors these, let's say, PAMPs from viruses, and they push the production of type 1 interferons. Type 1 interferons, they go in and they really kind of, let's say, push the production of ISGs. So what do these ISGs do? The ISGs basically, these interferons, so what do these interferons do? When interferons, when they go, they start binding to many different types of receptors. They could be their own receptors, so autocrine, or other basically cells. These are paracrine. So what they do is once they bind to the interferons, the alpha and beta, they really put these cells into some sort of way, this kind of alarmed state. They get alarmed, they start to produce a lot of these ISGs, as we said, these certain proteins that are there stopping replication in general. But the cool thing about this is that, okay, let's imagine that you have this infected cell. This infected cell now pushes the PRRs to produce interferons. Interferons are released and they bind to other basically cells. So these cells now, they are stimulated with basically of their ISGs, but they produce, so these basically proteins that are being produced, they're not active. They're there, but they're not activated. They will only get activated once they are literally binding to a virus, once their PRRs are activated. So they're just putting them in a steady state. They're ready, but they're not doing anything. So the proteins, these ISG genes, are now basically transcribed and translated into proteins that are ready to attack. But then they will not attack until a virus is literally stimulating their PRRs. So this one, basically, the ISG genes and the products are now really stopping replication because there's a virus. Here, now let's say you have your interferon over here. Your interferon's coming in, binds, it's now stimulating ISG, and it is literally producing a lot of, let's say, proteins that are stopping translation, but they're inactive. So translation and DNA replication is occurring because there's no viruses. In this one, the basically the interferons are binding, and then you have now your ISGs. They're not active until now that suddenly there's a virus. It starts to basically get activated, and they will literally push. So this is extremely important because this is really, it kind of helps this kind of, what it doesn't want to do with interferons is that what you don't want to do is making literally uninfected cells start stopping their activation or their replication or translation of their proteins, because that could be a waste. You're killing basically bystander uninfected cells, but you don't want to do that. That's toxic. So you want to be careful with that. So what are, let's say, these ISGs? What are these ones? What are these proteins? Well, there could be basically many different ways. The first one that I really want to talk about is activation of let's say, production of this enzyme called 2' 5' oligoadenylated synthase. So what this does, it literally synthesizes short oligoadenylated molecules. So these are these 2', 5' oligoadenylated molecules. And what they do is they literally will bind and dimerize together, and they will literally start binding specifically towards your RNase L. So RNase L what it does, it literally starts to cleave any RNA, any single-stranded RNA. So it stops both viral and host single RNA, because as we know, no matter what happens, what virus you have, be it DNA or RNA viruses, they will always have a single-stranded RNA that is ready to be translated. So what happens is that your ISGs, they produce this 2,5 oligodenylated synthase that produces this 2,5 oligodenylate nucleotides or molecules, which are literally causing this RNAs to dimerize and now to be active and start to cleave RNA. Another thing that can happen with these ISGs is specifically activation of protein kinase R. So these ISG, the products, these proteins can start to bind basically they can basically produce protein kinase R. Protein kinase R, what it does, it will bind to double-stranded RNA. So we know that double-stranded RNA is extremely found in viral infections. And once basically it binds, it's activated. That means that basically it's now ready to start attacking. And this PKR, once activated, it starts to phosphorylate initiation factors, and specifically this initiation factor 2 alpha. Initiation factor 2 alpha is really needed for translation. So what you do is you're literally inhibiting this eukaryotic initiation factor 2 alpha by phosphorylating it, and that now causes it not to work anymore, so translation cannot occur. So this stops both host and viral protein translation. So this can really help a lot in some sort of way, kind of your host cell from doing any viral replication or host cell protein translation. So it kind of stops viruses from replicating. So one thing that is quite interesting is that interferons that we just spoke about, they're quite actually selective in some sort of way. Specifically at a cellular level, interferon-induced viral proteins, they remain inactive unless triggered by viral replication intermediates. So even though the interferon-induced viral proteins, these ISGs that now are proteins, they're not activated. Even though they're produced, they're not kind of starting to do work. Only when there is certain, let's say, viral replication intermediates like double-stranded RNA. So for example, in this case of protein kinase R, even if it's activated, it's not going to start doing anything until there is, in some sort of way, double-stranded RNA. If there's double-stranded RNA, it's going to bind to it, and now that's going to be active. So that's how it happens. Same thing goes actually with this 2,5 oligodenylated synthase. The synthase will only get activated if there is a double-stranded RNA. If there is, then now it starts to really produce RNA-seq. So at a cellular level, these interferons are very specific. But at an organelle level, we see that they're also specific, meaning that interferons, they display some sort of specificity towards species. Meaning that if I take dog interferons and inject it to myself, they're not going to do anything. So in some sort of way, it's quite specific towards the species kind of level. And this is mostly because the receptor is different. The interferon receptors are very different among a dog or a human or a cat. So this is very important because we do create a lot of new drugs using type 1 interferons. But if we want to create a drug, let's say for humans, we need to make sure that this interferon is being produced from a human DNA, not from a pig DNA or something else. So most of the times that we do it, we use genetic engineering rather than extracting it from animal sources. Another very important cytokine that gets released is interleukin 6. Interleukin 6 is extremely fast. It's usually produced one of the most first cytokines along with interferons type 1s. They're produced by many different types of cells, specifically macrophages, dendritic cells, endothelial cells, and infected stromal cells. They basically are produced when PRRs are being activated. And interleukin 6, what it does, it really is very similar to your interferon gamma. Interferon gamma, it's mostly produced from other cells, but interleukin 6, it it usually, what it does, it actually pushes the activation of your adaptive immune system. So it really kind of bridges your innate and your adaptive. And how it does that, it actually produces or pushes B cell differentiation to produce antibody producing plasma cells. So it pushes B cells to become plasma cells. It also pushes T cells to get activated and survive. Specifically, pushes T cells to become cytotoxic. It pushes them to become T helper 1 cells. So we know that in your CD4 T cells, they can produce many different subtypes. In viral infections, we want T helper 1 cells. Those are the important ones that are really producing or helping viruses to be stopped. And they do this specifically through the activation of the JAK-STAT3 axis. It's extremely important for T cells to keep being alive and proliferate into T helper 1 and also cytotoxic T cells. But the issue with interleukin 6, even though it can be very, very, very protective and really can, let's say, kill or let's say kind of activate a lot of antiviral immune responses, it can be very pathogenic. It can be extremely at high amounts, and if it's systemic, it can really cause a lot of damage to surrounding cells. So this basically people that have a lot of interleukin 6, they have a lot of tender and swollen joints, joint destructions, a lot of hemoglobin issues, a lot of osteoclast activity, so their bones are being basically destroyed from the inside. It's quite a lot of stuff that can happen. But also high levels of interleukin 6 can cause your cytotoxic T cells really to get so activated that they become impaired. So they become actually exhausted and they cannot do any more work. Interleukin 6 can also reduce T helper 1 polarization. A bit of it can push them, too much can stop and start skewing towards other T cell responses, towards mostly T helper 17, towards other T helper cells that are actually they're not really needed for viral infections. So it's very dangerous having too much of it. Another very important cytokine that gets produced is TNF alpha. TNF alpha is strictly produced by macrophages and monocytes. They really love to produce TNF alpha, and they really produce a state of basically activation, specifically through this NF kappa B basically expression or transcription factors. So what they do, they can have a very potent direct viral effects, meaning that they can actually go into virally infected cells and they actually signal to apoptosis. They can also start recruiting neutrophils and macrophages to infected areas, which are super important for basically removing the pathogens. So they're very, very important. But of course, having too much TNF alpha can cause excessive inflammation, which could lead to pneumonia because you're bringing too many macrophages and neutrophils to your lungs that can cause pneumonia in your lungs. They can cause pulmonary edema because too much inflammation produces a lot of your lymph nodes start to release a lot of lymph, so that can cause edema. You can have a lot of tissue damage, so this is dangerous. And we see this a lot in SARS-CoV-2 and influenza. These are very respiratory viruses that induce TNF alpha production, which can cause a lot of macrophages and neutrophils to come into your lungs to do a lot of killing in there. Another important cytokine is interleukin 7. Interleukin 7 is usually produced by T helper 17 cells, so a subset of CD4 T cells. Produced also by NK cells and also CD8 T cells and gamma delta T cells. And they really start to be produced specifically when they're activated. And interleukin 7 is extremely important because what it does, it really pushes a lot, basically the production of certain chemokines like chemokine 1 and CXL8. These bring neutrophils in to do a lot of inflammation. But what they also do is they strengthen the barrier integrity. So they really help a lot of this endothelial barriers to kind of keep intact and not let viruses spread quite a lot into the blood. So it's super important for that. But so it mostly, let's say, works towards the barrier integrity, but also neutrophil. They're bringing into neutrophils. But having too much in neutrophils can be very, very dangerous. So too much interleukin 7 can really produce a lot of neutrophils to come in and do a lot of activation of releasing a lot of cytokines that can be very toxic. Another extremely important cells when it comes to viral infections are natural killer cells. So natural killer cells, what they do, as we said, we've spoken about this before, is they love to patrol and look at your healthy cells. Because usually in a normal cells, they basically all nucleated cells have MHC1. And that's the normality, and they keep basically presenting many different types of antigens. So NK cells, if they recognized certain, let's say, basically, let's say normal cells with an MHC, that by itself kind of says like, hey, this is a normal cell and nothing really happens. But as we know, and we've spoken of this about before, viruses, what they love to do is they love to stop the upregulation of MHC1 in the cell membrane, in the cell, basically cell and the plasma membrane, sorry. So they try to snatch it down. So in infected cells, for example, here you have this HIV in infected cell. So this is a cell that's not infected with HIV. You do not have anymore this MHC1. So what happens? This already kind of triggers your NK cells to kill, because when you have an MHC here, when you have an MHC, this kind of activates something called a killing inhibitory receptor, a KIR receptor. So this kind of inhibits killing. So the name says it. So when you don't have MHC, this KIR is not activated, and this pushes an NK cell to start killing. So viruses might think that they're being smart by removing MHC, and by removing MHC, you stop CD8 T cells to kill, but now you're making yourself vulnerable to be killed by an NK cell. So it's quite important to see how these interactions occur. And as we know, natural killer cells are extremely important when it comes to cytotoxicity, because they can kill through multiple different ways. So we know that they can kill through this fast trail. So we have this fast ligand, let's say, interactions. And when this happens, they really start to release granules, specifically perforins, which perforate the membrane, which release granzymes to go inside, and granzymes will start inducing apoptosis inside a cell. But of course, other than that, NK cells can really release a lot of different cytokines and chemokines, specifically chemokines that bring dendritic cells to come in, which will do a lot of antigen presentation during viral infections. So they're very, very, very important. Other than just killing, they really also try to bridge your innate system to your adaptive by bringing dendritic cells to come in to start doing antigen presentation. They can also bring a lot of macrophages and monocytes to come in and also do phagocytosis to induce MHC or antigen presentation. Another very crucial cell that is really needed for viruses is macrophages. Macrophages is usually the one that is actually phagocytosing viruses. Usually bacteria are phagocytosed by neutrophils. Viruses are mostly done by macrophages. They're very important, so they come in and they really start to phagocytose and do a lot of antigen presentation. But other than that, macrophages, I see them as literally kind of the conductors of an orchestra, because they really conduct and orchestrate immune responses, because they release so many different cytokines and chemokines, and examples interleukin 6 and TNF alpha that I just said, but they can also produce a lot of different types of cytokines and chemokines. Examples range from chemokine 9, chemokine 10, which bring NK cells. They can also bring T cells through chemokine 9 and 10 as well. So they really try to orchestrate immune responses by bringing them to the site of infection. So other than just phagocytosis, they orchestrate immune responses, specifically when it comes towards viral infections. They can also produce a lot of interferons that are basically, helping dendritic cell and T cell activation as well. Another thing that we see from immune cells that they produce are these alpha defensins. So alpha defensins are small, positively charged antimicrobial peptides. And what they do is they interfere with the entry of host cells. So this could be basically by binding to viral particles or by basically binding to the host cell receptors. So they don't have to basically do one or the other. It's one of them that basically stops viruses from entering. I'll give you a very nice case with HIV. In HIV, we can see that basically we have these alpha defensins that are produced by neutrophils, and what they do is they bind to chemokine receptor 4, which basically is a very important receptor for HIV. So by binding to it, it blocks it. So HIV doesn't bind to chemokine receptor 4, so it doesn't able to go inside. But also, other than basically that, we can see that host defenses, they can also act basically in the viral life cycle, but also in the entry and replication. But other than that, of course, then so what I mean, by the way, by this is that not that do they only basically bind to the receptors, they can also come in and they can also bind to other, let's say, viral proteins. They could bind to basically for example, in the case over here, you have this beta defensin, a different type of defensin that can stop basically there's reverse transcriptase enzymes. So they can actually have another, let's say, blocking the uncoating by blocking certain, let's say, membrane fusion proteins that allows this uncoating to occur. So the alpha defensins, or these defenses in general, can stop a lot of different things. Alpha defensin mostly inhibits viral entry, but you have, for example, beta defensins that can also bind to other different things. We also see that alpha defensins, they also have antimicrobial properties. So it's just to show that they're not just towards viruses. Another very important, let's say, protein that gets produced within our cells is this APOP ECG3 basically enzyme. This is a cytidine deaminase enzyme, and it really works extremely well with retroviruses, viruses that use reverse transcriptase, because what it does, it really binds towards reverse transcriptase, and what it does, it really kind of introduces this uracil. It makes basically so it converts uracil. It can convert cytosine into uracil. And what it does, it really puts a lot of mutations. So now this viral genome is non-functional because you have so many mutations that really stops a lot of proteins being expressed. So this is what APOC C3G does. And it really binds a lot to reverse transcriptase. So this really mostly works specifically towards retroviruses. That's where it really works. However, we see certain retroviruses like HIV that have, let's say, evolved certain countermeasures, like VIF, this viral infectivity factor. This VIF, what it does, it targets this APOP C3G for degradation. It really binds to it and it pushes it to be basically, let's say, killed. So this is just to show how the viruses are evolving certain strategies to combat our own strategies to stop it from replicating. Another thing that we see that is, let's say, antiviral is fever. Fever actually really enhances a lot of antiviral defenses. This could be, for example, elevating our body temperature. This really can have an effect in viruses because viruses need a certain temperature to replicate. So if you push the temperature, you can stop replication. You can also be very sensitive for enveloped viruses, because we know enveloped viruses, their lipid membrane is sensitive towards heat. So having high fever can really be pushing towards that. It can also enhance your immune system to start be more mobile and increase the activity of the leukocyte by being much more releasing of granzymes. So in some way, it has multiple, let's say, factors, but really the precise contribution, they really, let's say, 100% what we know how it's able to really kill viral clearance. It's not really 100% understood. We've seen that these things are elevated. These could help, but we have not really seen a very specific mechanism to see how fever really modulates. Another thing that we also know that is extremely important is the mucus layer, because one thing that we know in respiratory viruses, they love to go to our epithelia. But what we see within our epithelia, we have this mucus layer, and this mucus layer really stops viruses from going in. It's this very thick substance that does not allow viruses to just go to penetrate. But within this mucus, we know that there's a lot of different types of, let's say, antimicrobial peptides, but also antibodies, specifically IgA, that stop and neutralize your viruses. So we have these very important mucus layers that are really stopping or like kind of electrostatically or sterically inhibiting viruses from getting inside our epithelial cells to do destruction. And we see this a lot in people that smoke. When you smoke, you really kind of stop a lot of this kind of, let's say, epithelial, basically a lot of this kind of mucus. You remove a lot of this mucus, but you also remove these cilia, these very important small little hair-like projections that are really just clearing out viruses. They kind of stop viruses from let's say, infecting us. So we see these people that smoke, they have reduced mucus. You have a lot of basically this kind of the cilia being removed. They do not move. They're not produced that much, and you see these people having a lot of let's say, respiratory virus infections. So we also see that, of course, with our host defense, you have levels of immunocompetence. So we see that, of course, babies they do not have antibodies, and their T cells are not the greatest. So what happens is that usually, of course, babies are much more infected with viruses compared towards, let's say, a teenager, because they have certain, let's say, their immune system is not the best. But we also see that in older people, with older people, the thymus gets smaller and smaller, so T cells are not being really produced that much. But also you see with T cells and B cells, they go something called senescence, which basically means aging. Their T cells and B cells, they're not so active because they're quite old. So these people also are very susceptible to viral infections. But we also see some people that are, let's say, having corticosteroids. These are basically drugs that suppress your immune system. You see this with people with transplants. They really start to they have a lot of viral infections. Nutritional is super important because we know having deficiencies in proteins or micronutrients can really inhibit immunoglobulin synthesis, so you have less antibodies being produced, reduced phagocytic efficiency, and you also compromise your epithelial barrier. So really how basically your defenses are, being by age, being by certain drugs you take, by your nutrition, by smoking, this can really contribute towards how your body fights infections. So now we really did speak a lot about the innate immune system, but let's more speak now about the adaptive. So as we said, your adaptive immune system, specifically your B and T cells, they are very specific. They are trying to be taught and to basically target specific epitopes, either basically, coming from the virus. Most of these epitopes coming from the viruses. So they're trying to be extremely specific. So we know that our, let's say, adaptive immune system is divided into your T cells. So these are more, let's say, your cell-mediated arms. And these could be also, we have the humoral response, which is more your antibody. So your B cell response. CDT, basically your cell-mediated arms. So these are your cell-mediated immunity, your CTLs, your cytotoxic T cells. Your cytotoxic T cells are extremely important in eliminating cells that are already infected. These are important for that. CTLs will not kill a virus that is outside in your blood. It doesn't do that. We know CTLs will only recognize epitopes presented on MHC, specifically MHC1. While when it comes to the humoral response will only be able to kill through viruses that are outside. So these are important because B cells kill, or let's say neutralize, viruses that are found outside, while CTLs will neutralize viruses that are inside a cell. So different, let's say, locations they're basically killing through. So you need to think about CTLs where you, let's say, imagine where are CTLs very important is specifically when you have a viral infection that are already present inside your cell. Antibodies that are useless, you don't need them, so it's mostly you're relying on CTLs. But if basically the viruses have not yet reached inside the cells, your humoral response is much more needed. So as we said, active immunity really is coordinated by the activation of your B cells, your CD4 T cells, and your CD8 T cells. So we know that in specific, our innate immune system has now tried to stop as much as they can the viral titer. They start to come in, they start to literally bring the antigens towards the B cells, towards the T cells to start, kicking up your cytotoxic T cells and your antibody titers. So one thing we see, you have different durations. So what we see is, for example, in systemic infections like measles, we see this strong, long-lasting immunity, where we basically can have a huge amount of T cells, and you have a very good adaptation of a memory response. So once we're basically infected by measles, getting infected again is quite hard because we have a lot of memory B cells and memory T cells. That what happens is that when we are infected the second time, rather than taking 7 or 10 days, it takes 1 or 2 days. So you're stopping the virus from reaching this high titer already. So this is very important. However, what we see also with other, let's say, kind of infections like rhinoviruses, we can get infected 3 or 4 times a year. That's purely because we have very little antigen presented, it's short-lived, it's localized, you don't have a lot of T cells or B cells being formed, and the memory responses are not the greatest. So it's just to show that how are T and B cells being formed, it really depends on the viruses that we're being infected. Are they really causing a very nice T cell response, like specifically memory responses, B and T cells? This mostly happens. Are the antigens being presented very well? Is it more systemic? This really helps a lot because this can determine, will you ever get a vaccine, basically a infection again? And this really tells you why sometimes we do some viral, for example, with measles, when we get vaccinated once, that's it. We don't need it anymore. But with certain, let's say, viruses, we need basically to be basically given multiple ones. This example is hepatitis. Hepatitis B, we need 3 vaccinations because 1 is not enough because it's short-lived. It doesn't get presented that well. But having it 3 times really pushes our T cells to create a very great memory response. Same with your B cells as well. But one thing that we start to see specifically that I want to show that is, let's say, quite interesting. So B cells have a very important, basically, very, very crucial and indispensable role in helping us in secondary infections. Because during the first infection, specifically towards the first one, our antibody titers, like when they really are high, most of our viruses are really down, because most of your cytotoxic T cells are doing the killing. But these antibody titers are very, very important, specifically your memory B cells, because when you're infected, again, what really prevents viruses from doing a lot of damage are your memory B cells, because now they're literally causing a lot of production of antibodies against the virus that it's going to prevent it from infecting, because the cytotoxic T cells will only be able to do killing only when it's inside a cell. But in the case that where you have viruses now in your blood or in certain sites, it's really your B cells that are doing or stopping a lot the killing or the infection technically to spread. So B cells are extremely important for basically reinfections. So we see this a lot in vaccinations. We see, for example, what we see is that location matters, or specifically with antibodies. Because we started to see specifically during the pandemic, of the COVID pandemic, we started to see that people that if they had IgA antibodies, IgA, against SARS-CoV-2, they had a better protection. IgA antibodies, as we know, they're found in your mucosal sites. IgG is mostly found in your blood. SARS-CoV-2, this virus really infects mucosal sites. So IgG antibodies, even though you have a lot of them, they're not gonna really do much protection because the virus is infecting our epithelial or mucosal sites. So IgG, before it reaches, let's say, the blood, it's going to be already in a cell doing a lot of damage. We started to see. So you see here, basically, these are patients. These are people that had no infection, so they had very little amount of IgA antibodies against SARS-CoV-2. But then you see these are people that are basically now infected or had a previous infection. Now you see that they have a high titer of IgA antibodies against basically SARS-CoV-2. So what we see is that if basically people had IgA antibodies against SARS-CoV-2, they have basically less viral replication. So they had basically, so this is the relative risk of breakthrough infection. So they had less breakthrough infection compared to people that had basically, for example, IgG. No one really cares for IgG antibodies. It's very low. IgA is extremely important in mucosal sites. And this, of course, they started to see this really stopped viral replication. So this is the CT value, is basically qPCR. So having a high CT value means that there was less basically viral genomes. So they saw that the location matters. Where the antibody is located is extremely important. And it makes sense because, okay, the virus is first entering through your respiratory. You have an antibody there, like IgA. You're able to stop it from entering the cell. One other thing that we started to also really see that's super important actually is isotypes. Other than, of course, antibodies being neutralizing, what antibodies are extremely important is they actually have to do other effector mechanisms. So we know that there's neutralizing, and most of the antibodies except IgD and E, they neutralize. IgM, A, G1, all of them neutralize. Basically, most of them also opsonize, but when it comes to actually killing through NK cells, it's only select few, or activating your complement system. And specifically in IgG antibodies. So we know IgG antibodies are extremely important in inducing your immune system to kill. So IgG antibodies within our, let's say, humans, there's literally 4 isotypes. There's IgG1, IgG2, IgG3, and IgG4. So yes, I need you guys to remember this, to memorize this, that there's 4 isotypes of antibodies in humans. 1, 2, 3, and 4 of IgGs. IgG1 and IgG3 are extremely good at activating NK cells and also your complement system. IgG2 and 4, they're not. They're very bad at activating your NK cells. They do not at all, actually. And they're very poor at activating your complement system. So they don't do that well. One thing we started to see, specifically these are coming from HIV vaccine clinical trials, we started to see is that people that were vaccinated with this vaccine, some of them were actually shown to be protected while others didn't. But why was that? Despite that all of these participants had neutralizing antibodies against HIV, some were more protected than others. But why was that, despite them being neutralizing? Well, what people started to see is that their compartments of IgG really was different. So the types. This specifically came from Amy Chung. Amy Chung is a very cool scientist coming from the Doherty Institute, Research Institute in Melbourne. I met Amy Chung at many different conferences. I really wanted to do my postdoc in her lab. I was super close to go to her lab, but then COVID came in, and then Australia had stopped all basically the doors. I was very close, kind of still, to the degree I still, if I have the opportunity to go there again, I will definitely be going to Amy Chung's lab. Because what she saw is that patients that had IgG3 antibodies more than IgG4, they were more protected. Because if you look at IgG3 is very good actually at complement activation, but also NK killing. Very basic. IgG4 really doesn't do that much at all. It's actually one of the poorest antibodies. It's just there for neutralizing. And of course, these are basically 2 different types of vaccines. There's RV144 and VAX003. So these are patients giving these 2 different vaccines. So when she depleted IgG3, when she removed IgG3, there was no phagocytosis. And then when she removed here, basically IgG3, there was no specific lysis of infected cells. So no NK cells being basically killed at all. So when you removed IgG3, there was no phagocytosis and there was no NK cell killing. When you removed IgG4, nothing really happened, honestly. Even better. In the case of VAX03, there was more phagocytosis and there was much more actually killing of NK cells. So they started. She saw that people that were protected had certain antibodies, specifically IgG antibodies, that were very active. It didn't matter if they were neutralizing. They were actually, once they were bound towards a virus, they could actually induce killing through NK cell killing or through phagocytosis. And that was a form of a very important protection, much more important than just neutralizing it, actually. And this is now, Amy Chung now does this quite a lot for many, many different viruses, for dengue viruses, for SARS-CoV-2, for influenza. Her work has been a massive pillar in for us to understand how can we make a vaccine much more protective. Because now you want to create a vaccine that is able to produce certain, let's say, isotypes. For example, IgA, if you have respiratory viruses. If you have a virus that's more in the blood, and that neutralizing is not just super important, but you need, let's say, certain effector mechanisms like phagocytosis or NK cell killing, you need a more IgG1 or IgG3 kind of isotype. So really her work, be it as basic, when it comes to basic fundamental sciences, she really now helped a whole career, or basically, let's say, kind of field to come out to start saying, okay, the new vaccines have to be of a certain isotype. So now we have basically, of course, antibodies being, other than, of course, have the effector mechanisms, they are also very important in neutralizing. So the neutralizing really basically is very important to see about the epitope, accessibility, affinity, and stoichiometry. What that basically means is that antibodies, they bind to an epitope. If that epitope is accessible, it's going to bind to it. But if it's not, then it's very low chance that it's going to do anything. Despite that if this binds to here, it's going to stop the basically viruses from infecting, from infecting cells, because you're stopping the host receptor to viral receptor interactions. So the accessibility is extremely, extremely important. But also one thing that's super important is stoichiometric thresholds, meaning that there's a minimum number of antibody molecules that must bind to black infectivity. So as we know, viruses, they will bind to host receptors. They do this because of their own viral receptors that are found on the outside, specifically that are in the capsid. So let's imagine that technically it needs 5 receptors. If 5 receptors are basically binding towards the host receptor, and that's going to cause it to go inside, okay, that you need 5. But we know that the virus has, let's say, 10 receptors. If you're basically now have literally, let's say, 3 antibodies binding to it, there's 7 that are still accessible and can do infection. But if you have now 6, you only have 4 available, so now you're not able to. And that's stoichiometric thresholds. It's the minimum number of antibodies needed to block infectivity. And this, of course, it all comes to high affinity. So having high affinities really kind of helps the stoichiometric thresholds. But of course, one thing we start to see is antigenic drifts. These are happening with basically influenza viruses or other viruses like HIV, in where their receptors, specifically their epitopes that their antibodies are binding, they keep changing. So this stops the antibodies to bind. So it's very, very important. So neutralizing, basically not all antibodies can be neutralizing, some can be. So antibodies that are neutralizing means that they're literally binding to a specific protein in your virus receptors, and this binding stops the receptor binding. So it prevents literally the virus receptor from binding to host receptors. So that's very important. So as we know, there are certain, let's say, for example, And this is a neutralizing antibody. This is an antibody that's stopping, for example, this SARS-CoV-2 receptor, this S1, from binding to the ACE2 receptor. But you have a lot of receptors that are not binding, that even though they're literally binding towards the S1, it's still able to infect. These are called non-neutralizing antibodies. They still bind towards the receptor, the viral receptor, that is responsible to binding towards the host receptor, but it's not really interacting or kind of disrupting them from interacting. However, they're also super important. They become very underrepresented. People used to think that only neutralizing antibodies are important. No, as I just literally explained, having these non-neutralizing antibodies, they can really do a lot of other stuff. They can do phagocytosis. They can also do complement activation. They can actually cause NET, the neutrophils to start releasing NETs. They can also do basically a lot of cytotoxicity. So one thing, as we know, and specifically that there's a limitation with antibodies, that they are not able to go inside a cell. So virally infected cells, they are useless when it comes to an antibody. Antibodies will not be able to do anything towards it. So viruses exploit that. So what they do, they will start to spread between adjacent cells via membrane fusion. So the syncytia formation that herpes does, it tries to evade your antibodies from doing anything. So that's very, very important. But as we know, sometimes the antibody virus complexes can actually dissociate and variants can still remain infectious. So antibody and virus complexes, sometimes what happens is that it's not a permanent interaction, meaning that it's gonna dissociate, so the viruses will still be able to be released. But also what we see, specifically in non-neutralizing antibodies, specifically non-neutralizing antibodies like IgG 2 or 4, that they do not do anything over here, they can be very dangerous because they can also basically further deposit in tissues, and they can be very basically immunogenic in some sort of way. One thing that we also see that with antibodies is that other than being protective, they can also do something called AID. This is called antibody dependent enhancement. Sometimes, for example, if you have, let's say, virus particles over here, they literally can bind to them, and you create these massive immune complexes, which can be taken up by host cells, because host cells, they can actually also have, in some sort of way, Fc receptors. They can be taken up, and you're literally now pushing viruses to be actually taken up even faster. This is called antibody-dependent enhancement. So you're enhancing a virus to be literally uptaken by a host cell or even an immune cell, and in fact even better because of the antibodies. You see this a lot with dengue, and at one point we also saw this with SARS-CoV-2 viruses. So you really are pushing quite a lot, much more their infectivity. So antibodies being very protective, they can also be sometimes dangerous, specifically in dengue viruses, or specifically dengue, I would say. And another thing that we also know is super important is your cytotoxic T cells. Super importantly, their timing, their efficiency, and their tissue impact is extremely tightly also important. Because we know T cells, once they're basically, we have an dendritic cell presenting through your MHC1, it teaches them. Now you have a nice cytotoxic T cell, a CD8 one, that is able to basically kind of recognize virally infected cells through the MHC1, and this can really reduce a lot of killing. Usually, basically, this MHC1 interaction, but also through the Fas and Fas ligand pathways as well, releasing a lot of interferons, but also basically granzymes and perforins to kill. So they're very, very important when it comes towards virally infected cells. But one thing that we start to see a lot, and specifically in chronic infections, during chronic infections, what we see is that the viral antigens are exposed so much, but because they're exposed so much, our T cells start to become tolerant. They start to see it more as self, because it's been present so much there that your T cells start to realize, like, maybe this is just a normal cell or a normal protein, because it's always there. So you can stop your T cells from doing anything. But also, T cells, they become chronically exhausted. During chronic infections, when you have a chronic infections, T cells start to get super tired and they really start to become exhausted and they reduce their cytotoxic abilities and they reduce the amount of cytokines produced. So it's extremely important to make sure that the infections are not chronic, because this can be very kind of dangerous towards T cells. So as we know as well, of course, what also happens with our viral immune evasion is that we know that we have a lot of viruses that what they're trying to do is they stop a lot this MHC1 upregulation. So they stop antigen presentation completely. However, if they do that, we know that NK cells can come in and kill. But as we know, we have a lot of viruses that can stop interferon production. They can stop this by stopping the production of it, or by degrading, let's say, their receptors or the signaling, or they degrade the ISG, the kind of interferon stimulated gene products. They can also produce these virokines, these homologs of host cytokines, that they stop really cytokines from binding to the proper receptor. So there's many different ways that we spoke about how viruses can kind of stop or kind of evade your immune system. One thing that we also know is passive immunity. We know that basically we can basically pass on antibodies, specifically through, in some sort of way, naturally or artificially. So naturally, this is mostly coming from babies. So babies, we know they do not have B cells still yet. At the age, I think around 6 months, do they start to have their own B cells producing antibodies. So they're mostly getting their antibodies from their mother through breast milk. This is passive immunity, because you're giving them already antibodies. But we also can have artificial passive immunity. So we know that people that have, let's say, very strong viral infections, we can give them antibodies against that virus that they're infected with. This is more artificial ones. And again, it's super important to know what type of antibody. If you're having, let's say, literally mucosal, some sort, let's say, infections, you want to give IgA. If you have more blood-borne viruses or more in the blood, you want to be giving IgG. But of course, between passive immunity, antibodies have a certain half-life. Specifically IgA within a couple of weeks, 2 or 3 weeks, they're gone. So you need to keep supplying it. IgG, it's every 6 months. So around 6 months, you need to supply with IgG. One thing that is also super important with antibodies is they really help us with herd immunity. So what is herd immunity? Herd immunity really emerged when populations, basically herd immunity emerges is when population immunity reduces transmission probability. So this basically means that now, because you have a lot of people that are immune, they really will give you some sort of protection. So let's imagine you have 3 kind of people. You have people that are uninfected but unvaccinated. So these people have no infection. So let's imagine we have in SARS-CoV-2, okay? So in SARS-CoV-2, this patient has never gotten the infection and is not vaccinated. Then you have people that are vaccinated in blue, and then you have people that are in red that are infected. And people that are infected usually can pass on the virus. So what can happen? So let's imagine over here, you have basically a population that is that is mostly unvaccinated. What happens is that infected people are going to pass it on to each other, because they don't have some sort of response to kind of prevent your immune system. So these people do not have antibodies that are preventing the virus to come in. But let's imagine now you have basically a population where you have a lot of people vaccinated. So you have this infected person. This infected person is trying to infect as much as they can. So the people that it can really infect is this guy over here, this uninfected person. So what happens is that this infected person now, to get there, has to pass through all these vaccinated people. But it's really hard, because now when this person is releasing the SARS-CoV-2 particles, these people now have antibodies that are preventing the SARS-CoV-2 from doing anything, from literally going inside a cell and being effective. This is called herd immunity, in where you suddenly have a population that is reducing the transmission probability. And how basically the threshold depends on the virus's basic reproductive number. So that means a virus has a reproductive number, which basically means it's the average amount of times one infected individual can infect. So influenza sometimes has an infection, an R0 of 3, let's say, for example. That means that one person infected with influenza can infect an average of 3 people. Some viruses have a lower reproductive number. So if you have a low reproductive number, so let's imagine you have a reproductive number of 1, meaning that one infected person can only infect one person. Well, the threshold, how many people you need to have basically this herd immunity, is lower. Because, again, you're only infecting one person. If you have for example, in the case of certain viruses like SARS-CoV-2, which I think the reproductive number was 7 or 8, this person can infect 8 or 7 people. So you need a lot of herd immunity. So you need a lot of people to actually be vaccinated to protect the unvaccinated. So depending on the virus, you need a certain threshold of herd immunity. So how many people to be vaccinated? So usually during the programs of vaccination, people or countries were trying to reach, for example, vaccination rates of 80%, because they knew that at 80%, you have so much herd immunity that the rest of the 20% of people that are unvaccinated will be protective because of herd immunity, because they knew that 80% will cover the amount of, let's say, of the R0 of SARS-CoV-2. But of course, we also know that not all immunity contributes equally. So we know that in SARS-CoV-2, having IgA was extremely important, because IgA would stop any of the viruses from entering your mucosal sites. So if you had IgG, it doesn't really matter. So it's not just only on the R0, but it also depends on what sort of immune system you have. Super important. So this is the end, guys. If you have any questions, please let me know. Thank you.